Hello, how are you? In this video, I'm going to go over the lecture that I gave at the Center for Biological Investigations in Madrid, in Spain, on July 28, 2022. As you know, cadherin receptors are necessary for all aspects of tissue morphogenesis. E, which stands for epithelial cadherin, plays a role in epithelial mesenchymal transition. If E cadherin is lost, then the cell loses its polarity and starts to migrate and to become invasive. So, E cadherin is clearly important in tumor cell invasion and metastasis. And the reverse epithelial mesenchymal transition happens because of high E cadherin expression, which leads to cell organization and cell polarity. The blue cells do not express much E cadherin, and the yellow ones do. The differences in expression of E cadherin drives tissue separation during embryonic development. So the neuronal cadherin allows the neuronal epithelium to separate from the E cadherin expression ectoderm. So what fascinates me is that on the one hand side, cells have to break cadherin adhesive bonds to allow cells to move, and they also have to make cadherin adhesive bonds to allow cells to build tissues. Cadherin is important to develop the compartment boundaries in a tissue and to form the rhombomeres in the developing vertebrate hindbrain. This requires selective adhesion by selective adhesion strength. N-cadherin is important for the growth, cone motility. In the synapse formation, cadherins form adherens junction as part of the synaptic junction. And finally, cell migration. So apart from E cadherin stabilizing epithelial junctions, E cadherin also enables the long-range migration of cells through tissues. For example, the migration of border cells in the Drosophila egg chamber to the oocyte. I am really fascinated by the interplay and the role of actin within cell junctions. Actin is important in tight junctions and in adherens junctions, while desmosomes have desmosomal cadherin that are linked to intermediate filaments. So they connect the intermediate filament network across the epithelial sheet. The initial cell-cell contacts are formed by opposing cadherin beta-catenin complexes at the tips of filopodia, as well as lamellipodia. And then alpha-catenin connects to the actin cytoskeleton. This allows for bundles of radial actin cables to assemble and to anchor to the underlying cortical actin ring. And finally, actin reorganization results in mature adherens junctions. As you know, there are three types of adherens junctions in the punctum adherens. Actin filaments associate with the plasma membrane and are perpendicular to the plasma membrane in the zonular adherens. Actin filament bundles are parallel to the plasma membrane in the tricellular adherens junction. Actin filaments associate with the plasma membrane at a large angle. The linear adherens junctions are found in the interior of a colony, and punctate adherens junctions are found near the edge of the cell colony. How actin filaments are assembled is important for the function. And here they are perpendicular and terminate at the plasma membrane. And this is important because they pull on the cadherin. In classical adherens junction, has the extracellular regions of cadherin changed the conformation. When binding to calcium, which leads to cadherin homodimerization, adherens junctions are stabilized by cadherin, which again binds to beta-catenin inside the cell. And beta-catenin then binds to alpha-catenin, which recruits many other cytoskeletal proteins. In the original view, alpha-catenin makes direct contacts with both beta-catenin and the actin cytoskeleton. Although alpha-catenin also interacts with cytoskeletal proteins such as vinculin and alpha-actinin. So the link from catenin complex could occur via vinculin or alpha-actinin also. Catenins also regulate the cytoskeleton by signaling. For example, P120 catenin can regulate the GTPases, Rho and RAC. Formin 1 binds alpha-catenin and regulates the assembly of linear actin filaments. And phosphatidylinositol 3 kinase interacts with beta-catenin. At the top, you see the clustering structure of cadherin, which brings me to the regulation of homophilic cadherin interactions. So cis-cadherin dimerization and higher order clustering is controlled by the phosphorylated catenin, where phosphorylation leads to deubiquitination and proteasomal degradation, which then severs the clustering. Alpha-catenin and vinculin are very similar, both structurally and functionally. They are both made up of mainly 4 helix bundle domains, where 2 such 4 helix bundle domains have one long shared alpha helix that connects them. Vinculin is larger and has 3 sets of double 4 helix bundle domains, while alpha-catenin only has 2 sets of double 4 helix bundle domains. Both proteins have an F-actin binding domain that is a 5 helix bundle domain. On the left, again, you see the original view where alpha-catenin makes direct contacts with both beta-catenin and the actin cytoskeleton. The middle domain, M1, binds to vinculin and is a folded 4 helix bundle. But the vinculin binding domain, M1, is buried, and the other middle domains, M2 and M3, are keeping alpha-catenin compact, and importantly, cytoskeletal tension releases the M2, M3 interactions, as well as the M1 4 helix bundle that binds to vinculin. So alpha-catenin M1 unfurls first to bind to vinculin, since they both have F-actin binding domains. It is not surprising that the alpha-catenin vinculin complex binds more actin filaments than alpha-catenin alone. Okay, so back to the linear and punctured adherence junctions. These are interconvertible in epithelial cells, depending on their cellular context. So again, actin filaments run parallel to the cell borders to form linear adherence junctions, which are also known as the zonal adherence. As I mentioned, in punctured adherence junctions again, actin filaments are perpendicular and terminate at the junction. So these actin filaments pull cadherin from both sides of the junction. So you have these actomyosin cables that generate the tension of cell junctions. As you know, there are 3 distinct alpha-catenin isoforms: alpha-E-catenin, alpha-N-catenin, and alpha-T-catenin, with different expression patterns. E for epithelial catenin is found everywhere, and N for neuronal alpha-catenin is found in neurons. And T for testis alpha-catenin is expressed in testes as well as in the brain and the heart. As you can see from the table, these 3 different alpha-catenins are very similar. They even have high similarity to the roundworm ortholog HMP1, which stands for humpback based on the phenotype that we'll talk about later. And I already mentioned the similarity with vinculin. As you know, lower vertebrae do not have the area compositor in the heart. The heart of mammals deals with an increased mechanical load by anchoring both actin and intermediate filaments over an extended junctional area of the intercalated disc. This is accomplished by plakophilin 2 binding to the intermediate filament and to alpha-T-catenin. Alpha-T-catenin then also binds the actin cytoskeleton. Okay, so why should one care at all about alpha-catenin? What is the major cause of death worldwide? Exactly, heart disease. Loss of alpha-catenin activates YAP and beta-catenin, which leads to cardiomyocyte proliferation and hypertrophy. This also leads to the activation of the PIP kinase AKT pathway via MAR29 and or LGFR1, and then PPAR alpha downregulation. All of this leads to the lower levels of fatty acid oxidation and to the accumulations of lipids and cardiomyocytes and possible cardiotoxicity. As you know, the intercalated disc of cardiomyocytes contains the desmosome, adherence junction, and ion channels. Mutations responsible for arrhythmogenic cardiomyopathy are found in 1. Components of the desmosome, 2. Components of the adherence junction, and yes, my favorite protein, alpha-catenin, 3. Contributors to calcium handling, 4. Intracellular structural proteins, 5. The sodium channel and transforming growth factor beta-3, and 6. Nuclear envelope proteins. Here's at least one other reason why you should care about alpha-catenin. What's the second cause of death globally? Exactly, cancer. And alpha-catenin is so much more than just a structural linker between cadherin complex and the cytoskeleton. I mentioned before that alpha-catenin also functions as a signaling molecule in the skin. Alpha-catenin is a tumor suppressor that senses the cell density. And I talked about alpha-catenin also being an upstream negative regulator of YAP1. So again, loss of alpha-catenin results in YAP1 dephosphorylation and activated YAP1 translocates then into the nucleus, where it binds TEAD, which activates hyperprofilation genes. So you have a massive overgrowth phenotype. When alpha-catenin is lost in the skin, as well as in the developing brain. Now, alpha-catenin can be a dimer or a monomer and has different functions in those two states. The problem working with alpha-catenin is that the dimer converts into the monomer and the monomer converts into the dimer. This Segmal's data show that when Rangarajan takes the alpha-catenin dimer, he still gets the alpha-catenin monomer and vice versa. When Rangarajan takes the alpha-catenin monomer, he still sees the alpha-catenin dimer forming. However, when he truncates the amino terminus, then he can isolate the alpha-catenin dimer. And for this truncated construct, the alpha-catenin monomer converts much more readily into the dimer. The last panel on the bottom on the right summarizes years of establishing a protocol to go from our original 9 angstrom diffracting crystals to about 3.7 angstrom. The structure that Rangarajan determined of alpha-catenin dimer is shown here, with one subunit in gray and the other subunit is colored spectrally. The two views are rotated by 180 degrees. In cyan, you can see the buried vinculin binding domain. The structure of alpha-catenin dimer has additional surprises. Although there's a twofold axis that relates the first 661 residues, the carboxy terminal F-actin binding domains are not related by that twofold axis. The cartoon on the right shows how the F-actin binding domains would be oriented if they were following the twofold axis that the rest of the molecule has instead. The second F-actin binding domain is flipped, as you can see in the cartoon on the top. Okay, vinculin, my second favorite protein, and here's why. Vinculin, like alpha-catenin, regulates cell-cell junctions, but it also regulates cell matrix junctions. And vinculin, like alpha-catenin, is embryonic lethal and a tumor suppressor. Vinculin is also important in wound healing and apoptosis. The crystal structure determination of vinculin was another nightmare. We had to develop a streak-seeding protocol to eventually get to 2.9 angstroms diffraction. Although vinculin is similar to alpha-catenin, both in terms of function and in sequence, vinculin is a monomer. And as you know, alpha-catenin is primarily a dimer. And unlike alpha-catenin, however, vinculin has a nanomolar head-tail interaction. And the tail domain in vinculin is also the F-actin bind domain, like in alpha-catenin, which is colored in gray. So the first set of double 4-helix bundle domains that are shown in blue bind to alpha-catenin. So basically, vinculin is autoinhibited and does not bind to F-actin or to alpha-catenin before vinculin is activated. I just told you that vinculin is autoinhibited and does not bind to alpha-catenin. However, in this native gel, you do see some complex formation, but it is a very small amount. When Rangarajan takes constitutively active vinculin by deleting the carboxy-terminal F-actin bind domain that severs the head-tail interaction, he sees the vinculin alpha-catenin complex forming more easily, and it forms with a 2-to-2 stoichiometry. When Rangarajan does a titration, he first gets a 2-to-1 complex of one vinculin molecule binding that then eventually saturates as a 2-to-2 complex. All of this opening up of both vinculin and alpha-catenin is not very useful to get a structure. Therefore, we want to find the minimal binding domains. The top shows the minimal alpha-catenin domain that binds to vinculin, and this 4-helix bundle is also a monomer or a dimer, as seen in our full-length alpha-catenin sample. The bottom segmals shows that we can obtain a 1-to-1 or 2-to-2 vinculin alpha-catenin complex, and the structure of full-length alpha-catenin is shown looking down a twofold axis onto the two vinculin bind domains of alpha-catenin that interact with each other. In the vinculin alpha-catenin complex structure, one alpha-catenin subunit is shown in gray as a cartoon, and the other subunit as a ribbon, and vinculin is colored spectrally. So when Rangarajan solved the structure of the vinculin alpha-catenin complex, he found a cysteine residue on alpha-catenin that forms a disulfide bond. So you end up with a 2-to-2 stoichiometry for the vinculin alpha-catenin complex. And the view on the right is looking down from the top of the view on the left. Okay, so back to my fascination with the cytoskeleton. And again, the original view of how alpha-catenin links adherence junctions to the actin cytoskeleton, which was long thought to be a direct interaction between alpha-catenin and the cytoskeleton. But then two papers were published in Cell that showed that only the alpha-catenin dimer, but not the alpha-catenin monomer, binds to F-actin. These groups are stellar and have produced stellar data over decades. But what has not been taken into consideration, in my view, is what I showed you earlier, that the alpha-catenin monomer converts into a dimer and vice versa. So in my mind, this monomer-dimer transition makes it very difficult to deconvolute the function of the monomer from the function of the dimer. There are a ton of data showing that force plays an important role in these interactions. The most recent view is that a slip and asymmetric catch bond occur between alpha-catenin and the cytoskeleton, and also a new interaction which I find interesting, has recently been identified between alpha-catenin and PIP3 in Philopodia. I was super fortunate to be able to install the first cryo-arm in the United States. This is a state-of-the-art 300 kilovolt cryogenic electron microscope. Now we no longer have to worry about these difficult crystallizations that we had in the past 2 decades. Rangajan was able to isolate the alpha-catenin monomer enough to be able to get the monomeric alpha-catenin cryo-EM structure. But alpha-catenin is such a flexible molecule that despite of leaving no stone unturned, we could not get more than 6.9 angstrom resolution. Rangajan was first disappointed to see in a structure that the amino terminal 2,4 helix bundle domains were disordered. But that flexibility actually tells us a lot about alpha-catenin function. It showed that the amino terminal domain is flexible and detached with no interdomain contacts. Also, the monomeric structure showed new secondary structural elements that we did not see in our dimeric crystal structure. For example, there's an alpha helix between the linker region and the F-actin bind domain. This alpha helix is potentially like a spring that keeps the F-actin bind domain closer or further away depending on the environment. Our structure is also consistent with published SACS data from other laboratories that showed such flexibility. So when we compare our monomeric cryo-EM alpha-catenin structure with our dimeric alpha-catenin crystal structure, we find that the F-actin bind domain has several orientations. I mentioned the 2 distinct orientations in our dimeric alpha-catenin crystal structure. These are shown on the right. 2 movies, colored spectrally. And we have an entirely new F-actin bind domain orientation in our monomeric cryo-EM structure, which is functionally important in terms of binding to other proteins. Back again to my fascination with the cytoskeleton. So Emmanuel then developed a protocol that allowed the cryo-EM structure determination of dimeric alpha-catenin bound to F-actin. The F-actin bound cryo-EM structure is challenging for many cytoskeletal proteins because they are either only partially decorating F-actin or they bundle F-actin, both of which is a problem for structure animation. But his perseverance paid off, and he got a 2.7 angstrom resolution structure, which is the highest F-actin bound structure ever reported. This is a Coulomb map from our cryo-EM structure showing the bound ADP and magnesium. As you know, it always helps to have high resolution. So this kind of high resolution detail was very helpful for us to have. So as with Rangajan's monomeric alpha-catenin cryo-EM structure, Emmanuel was also disappointed to see that in his structure, he could only see residues 710 through 871, but as in Rangajan's monomeric alpha-catenin structure, the flexibility actually provides a lot of insight into function. So most importantly, it means that the F-actin bind domain in the alpha-catenin dimer is readily available for binding to actin. This means that force is not required for the initial interaction with the cytoskeleton. The other interesting feature is the unfurling of the amino terminus of the alpha-catenin, F-actin bind domain, shown in red, which detaches from the F-actin bind domain and reaches over to another alpha-catenin, F-actin bind domain. This movie shows the effects on alpha-catenin when it binds to F-actin. The red arrow shows the changes in the last alpha-catenin alpha helix, which moves to the right, and the white arrow shows the release of the first alpha-catenin alpha helix, upon binding to F-actin. When we compared Emmanuel's F-actin-bound alpha-catenin structure with Rangarajan's dimeric alpha-catenin crystal structure, we see that in the so-called A subunit, the amino terminus, which is shown in red, is also detached from the F-actin bind domain, but it points in a different direction. So Emmanuel determined the cryo-EM structure of unbound F-actin also to 2.7 angstrom resolution, which is also the highest resolution reported for F-actin so far. Emmanuel's F-actin structure has reasonable temperature factors. The highest temperature factors are found for the so-called D loop. This loop has been known to be flexible and to adapt different conformations upon binding to different proteins. As you know, actin has 4 subdomains. The subdomains are arranged around the hydrophobic and the nucleotide cleft. And you can see ADP and magnesium in the center. On the right, you can see the D loop and how it extends into another actin within the filament. This little movie shows what happens to actin upon binding to alpha-catenin. In white is the unbound F-actin structure. In black is alpha-catenin superimposed, as we see it in the F-actin bound structure. And in yellow, you can see the transitions in actin to accommodate alpha-catenin binding. So back to the 2 seminal publications in Cell that changed the way the field thinks. We wanted to determine the function of monomeric alpha-catenin without the contamination of dimeric alpha-catenin function. So we chose HMP1, which is the roundworm ortholog of alpha-catenin that has over 36% identity and almost 60% homology with human alpha-catenin. And HMP1 alpha-catenin is a strict monomer that does not dimerize at all. So evolutionary, it is believed that HMP1 alpha-catenin diverged from the ancestral catenin complex, but it's unclear if HMP1 alpha-catenin, when it is bound to HMP2 beta-catenin and HMR1 catenin, shown in the red box, also binds F-actin readily. Emmanuel also determined the cryo-EM structure of HMP1 alpha-catenin bound to F-actin. The final resolution of the cryo-EM structure of HMP1 alpha-catenin bound to F-actin is 3.4 angstroms. And yes, of course, we have allostery again. What else did you expect? This little movie illustrates the changes that occur in HMP1 alpha-catenin when binding to F-actin. Upon binding to F-actin, tryptophan 711 moves out of its pocket to make room for tryptophan 859 to fill that space. This seems to extend the alpha helix H2 shown in blue and to stabilize the helical bundle. Here's my view of why alpha-catenin dimer binds to F-actin but not the monomer. It's as simple as bivalency. The alpha-catenin has high affinity simply because it has 2 binding sites compared to the monomer. And the other novelty from our structure is that alpha-catenin molecule has much looser interdomain interactions than previously thought. Certainly much weaker interactions than vinculin, which is autoinhibited. In addition to Rangarajan and Emmanuel being my heroes at the bench and at the microscope, I am grateful to NSF and NIH for providing the funds to make so much fun possible. Thank you for watching this video.